So in this particular talk, what we're going to deal with is osmosis, if I could speak. In particular, how the distribution of solutes affects where we find water inside of our bodies. So from last time, what we had was the cell membrane, at least at the very end, we had this cell membrane. And the job of the cell membrane, in part, is to regulate how diffusion turns out to work. Diffusion is just the movement of particles along a gradient. So along some type of slope, usually from a high to a low, in terms of concentration or something like that. For our purposes, we're just going to talk about concentration because it's going to be easier. So when we have this thing, proteins want to move from one side, or particles want to move from one side to the other, but this thing, this big old fat layer that we call the cell membrane, is going to be in the way. And because of that, it's going to slow down what gets to move around. If I actually look at that membrane, that double layer of fat, that phospholipid bilayer that you calculated out on the worksheet, what we notice is a few things. First is it's in a constant state of motion. It's not holding still. We like to think of a membrane, the outside of a cell, as being kind of a rigid structure, but it's not. It's actually constantly changing its shape. And the reason for that is fat molecules don't freeze that nicely. They don't lock in place, especially at body temperature. So what you end up getting is this fluid motion inside of the cell membrane. What you'll also notice is it's not just a bunch of phospholipids. There's other things like proteins and then stuff attached to all those proteins and attached to the phospholipids. And those things there kind of look like a bunch of broken pieces that are scattered all over. We give that a very specific name, and that's called being a mosaic. So the way that we try and model or describe, without actually seeing it, but the way we try to mentally give us a picture of the cell membrane is it's a fluid mosaic, meaning it's in constant motion and there's a lot of parts inside of it. One of the ways that we can demonstrate that is with this video. I'm not going to play the entire video I'm not going to play the video in this one. We watched it in class. You have a link to the video above in Canvas, so feel free to watch it there. But it's about 7-8 minutes long, and it actually is pretty good. I don't like the sound, so I always have the sound off. But it is really good at showing you the fact that the cell is huge compared to what you think it really is. The phospholipid bilayer is insanely small, and everything is in motion. So the way that the cell membrane turns out to regulate or control diffusion is it has proteins. Proteins are picky, meaning they will only do a certain job for a certain substance, and they will not change no matter what you beg of them. There are some particles that are capable of moving right through the phospholipid bilayer. We call that simple diffusion because there's nothing that's necessary. Those are things that can dissolve inside of fat because the phospholipid bilayer, lipid, is fat. Therefore, these are things that dissolve in fat. What would you notice that does this? Well, gases would do this, and fat particles would do that. Or anything that turns out to be non-polar, if you happen to remember what non-polar meant from chemistry last year. Things like acetone and methane, stuff like that. Other things that turn out not to be fat-soluble, they need help getting on through, and they need a protein. So we're going to need a protein to let these particles move on through. So what these particles can do, or the way that we can control these particles moving through around from one side of the cell membrane to the other, is I can either have it so it's a gate, meaning it can open and it can close, or it could be a channel where stuff can just dump on through and it just follows concentration gradients from high to low. So what we notice is there's some things that can move across the cell membrane. We don't need to help it. But other things like sugar, we need help. So sometimes we want the sugar we don't do this. We actually will control where it goes, and we use these things that we call gates. There are other things like sodium and potassium. We have times when we want sodium or potassium to be controlled when they're allowed to move, so we'll use these things called gates. Or we can want them just to move whenever they want, and we call that a channel. Both of these fall under the words facilitated. If you look under the B and the C, facilitated. What we mean by facilitated is the protein is there to help the motion. It's not adding energy. It's not directing. It's just saying, oh, you want to move? allow me to assist you. The far right side made reference to something called osmosis. And osmosis is the diffusion of water. And that turns out to be a very big deal, especially because in biology, living things are mostly water. That is the bulk of your mass. The way water turns out to move is it moves from high concentration to low concentration. The catch is you've never been asked to calculate water concentration before. So we need some tricks. So the way that we can view this is as a manipulation of diffusion. So diffusion, when we allow particles to move, either because we want them to or because we're forcing them to move, what you get, or what you could get potentially, is a buildup of solutes. So in this picture here on the left, what you have on the left-hand side are some sugar molecules, and on the right side there are more sugar molecules, however that happened. In between, we have what we call a semi-permeable membrane, meaning some things can move and some things can't. What you get is if I were to compare the amount of water as part of the total on the left-hand side, I will notice there's actually more water on the left than there is on the right. You say, but the heights are the same. My response is, that doesn't matter. On the right-hand side, there are more things filling up the space that turns out not to be water. The result is there's more water on the left than there is on the right, and water is going to diffuse from the left side to the right side. What would I notice? I will notice the picture on the right, which is water is actually going to flow from one side of this beaker to the other, and we're going to actually have a split in the heights. Is this something that happens? It absolutely happens. Because water is so much smaller than the phospholipid bilayer, if you remember from your worksheet, it's about 80 times smaller, it's possible for water to slip on through the cell membrane. It's not supposed to be, but it's possible. The problem is that's going to be slow. So in order to speed up this motion, there are proteins that are called aquaporins, and those help move water from one side to the other. So they're a protein channel that only allows for water to move. They will not allow sodium or potassium or chlorides or carbon or anything else that kind of looks like water. The only thing these will allow to move is water. This is kind of a fancy picture of it because it shows you a little bit of protein structure with these little spirally things. That's a far more stylistic version where we don't show you any of the fanciness of it. These funky things here that look like bobby pins or like a big alien with two tentacles, those are what we call phospholipids. We normally don't look at diffusion, or look at osmosis in terms of water moving. We usually look at it in terms of, well, what's going to happen? And then we use osmosis to explain what happens. So if I were to put a red blood cell, which is what that disky thing is, inside of a solution and it doesn't change its size, meaning I keep it in there and it doesn't change, it doesn't get bigger, it doesn't get smaller, what I say is that solution is called isotonic. It has the same strength as whatever is inside of the red blood cell, meaning the concentrations of stuff on the outside and the inside are the exact same. That's what we mean by isotonic. If I were to take a red blood cell and put it inside of a solution, and the solution is not as concentrated as the red blood cell, meaning there's more water comparatively on the outside than there is on the inside, the result is water flows into the cell and it looks like it's swelling up. And we call that solution, the stuff outside of the red blood cell, hypotonic. So isotonic, the red blood cell won't change. Hypotonic, the red blood cell gets bigger. So the only other option would be if there's more stuff outside, so there's more solute outside of the cell than there is inside of the cell. What that means is, comparatively speaking, there's more water inside the cell than there is outside of the cell. Water will then leave the cell, because water goes from high to low concentration, and the cell will shrivel up, or it will crenate. We call a solution that makes that happen hypertonic. So we have three options. We either have it so our cells stay the same, our cells swell up, or they shrivel up. The one that's best for life is the first option, the isotonic one. And that's because nothing adverse is going to happen inside of your cells. The way that we practice this is we look at some problems. I'm just going to post these here, and I'm not going to explain the answers. In class, we'll explain the answers so you can see if you can figure them out yourself. So you have a beaker with semi-permeable membrane. So we have that thing that will only let water move and nothing else will move. On the right side, there's 200 milliliters of water that has 100 milliosmoles of solutes. Whatever milliosmoles means, for the sake of saying it, you don't need to know what that means to answer the question. The compartment on the left has 200 milliliters of water and has 150 milliosmoles of solutes. Where will the water flow and why? If you draw a picture and you put some labels and you paid attention to the pictures in the past, you can figure it out. Another question. You have a cell that turns out to have 70 milligrams per deciliter glucose, or 90% glucose, inside of a beaker of water, and that beaker of water is 100 milligrams of glucose. Where's the water going to flow, and how do you know? Why do we care about any of this stuff? Well, diffusion osmosis is kind of a big deal, especially with us. We are eukaryotic organisms. And the only other term that you may have heard of is a prokaryotic organism from when you took biology once upon a very long time ago. And without going into the super nitty-gritty between these two, the major difference for our purposes between eukaryotic and prokaryotic cells, again, this is just our purpose, there's more than just this, is if I look at a eukaryotic or a prokaryotic cell, there's one membrane, which means I have two compartments. I can be inside or I could be outside, and those are my options. I have no other choices. If I look at a eukaryotic cell, I have a membrane on the outside, but then I have these other things like mitochondria and nucleus and endoplasmic reticulum and Golgi apparatus and lysosomes and stuff like that with all these funky words and vesicles. But each of those things is covered by a membrane. So what does that mean? I have a compartment outside of the cell. I have a compartment inside of the cell. But then I have a compartment that we call a nucleus. And I have a compartment I call mitochondria. Then I have a compartment that I call rough endoplasmic reticulum. And I have another compartment that I call smooth endoplasmic reticulum. And I have a compartment that I can call the Golgi. And I have a compartment I can call the lysosome. And I have a compartment I can call a vesicle. I have different places I can hide stuff. I have various locations where diffusion and osmosis can occur, which means I have far more control over the environment for the cell because we have places to hide stuff.